Hello, everybody. I'm Emma Blanchette. I'm representing Dr. Rehse's lab, and what we do in our lab is we use laser-induced breakdown spectroscopy to diagnose bacterial species. So in our introduction box, it details our process for collecting bacteria and the spectrum that's shown. So the process is given in the flow chart. We swab bacteria off a metal plate to simulate swabbing off of some infected surface. We then centrifuge all that bacteria and water onto a nitrocellulose filter, and we concentrate it onto that filter. And then we ablate that target in an argon environment. The result that we get is shown in the teal box. It is a lib spectrum. This is a typical bacterial spectrum that we see. And this overall process was created to mimic a clinical setting. At the bottom of the introduction box, you're going to see 19 lines from Optical Lib's spectrum. This is just a condensed version of our spectrum, and it highlights the lines that we look at in our analysis. So our overall motivation for this work is we want to be able to detect and classify the smallest number of bacterial cells possible. And we approach this problem from a few different angles using chemometrics. So if I can now direct your attention to the graph in the center, lib spectral intensity as a function of bacterial concentration. This is basically a graph that shows the normalized intensities of spectra that we obtain from a single shot. So separate species are encoded by color in this graph. Separate bacterial concentrations are given by the shapes within the graph. And sitting at the bottom here, the black dots represent a blank filter with nothing on it. The blue dots represent a filter with just DI water on it. This is important because we always suspend our samples in DI water. So we use this as a control, basically. And you can see from this large red circle or oval here that we have a huge amount of scatter in our data. We have points that have very, very high intensity at the top and very low intensity resembling that of water at the bottom. So this low intensity poses a huge problem for us when it comes to bacterial detection. Underneath this graph, we have SEM Images showing a non-uniform deposition of bacteria on the filter. And if you look closely, you can see little cracks in between the filter craters. As well, the image on the right shows voids between the bacterial lawns that is deposited on the filter, and we attribute this non-uniformity on the filter to our scatter in the data. So now for our results for this particular poster, directing our attention first to results, chemometric analysis, and algorithm comparison. All we wanted to do here was we wanted to see what algorithm was best for discriminating in between species. We did this by generating a well-behaved data set or pseudodata to test the efficacy of each algorithm and then compared the sensitivity, the specificity, and the classification error. A graph detailing the classification error results is shown at the bottom of this box. And during our analysis, we noticed that Staphylococcus and Enterobacter were often confused for each other. So we chose the classification error that minimized, or that was the smallest between these two. As you can see, the classification error for artificial neural networks, or ANN, and discriminant function analysis, or DFA, was very comparable. We ended up choosing DFA because DFA simply runs a lot faster on our machines and is more efficient for us to do than ANN. So now moving over to our results for the actual DFA classification of bacteria by species. This box basically just details our method of how we run this analysis and our results, which are shown in the bottom table. You can see that our sensitivity is quite low, but our specificity is quite high for classification in between species. As well, the 3D plot that sits to the right shows pretty good separation between groups. And finally, we come over to PLSDA detection of bacteria. So here we wanted to use partial least squares to determine the difference between bacteria and water. We just want to see if bacteria is there, basically. This particular analysis was run between water and E. coli bacteria. And first we started with trying to discriminate between single-shot Lib spectra. The results are shown in this table here to the left, and you can see that the sensitivity and specificity for the model was quite high, but when performing external validation on that model, the sensitivity decreased. Now, we know that when doctors are in the clinic diagnosing someone, they aren't going to want 20 to 30 answers, which is what this analysis gives us. They're just going to want one answer. So this is what motivated our next approach of just summing the spectra together. So that is given in this section here. The results are given in the table to the right. And you can see that the sum filters greatly improved the sensitivity and specificity and gave us 100% accuracy in this analysis. Below this box for PLSTA, you can see some figures in SEM image with a filter. This filter has about 10 laser shots on it. And to the right of that, you can see a spectrum. This spectrum illustrates how the summed or the add all given in the table above actually helps our discrimination. So here you can see the red spectrum or the single shot spectrum has a very low signal to noise ratio, whereas our blue spectrum or the add-all spectrum has a very high signal to noise ratio. As well, key lines in our analysis, such as magnesium and calcium, are greatly enhanced by this procedure. So in conclusion, our recommendation moving forward is to continue summing single shot spectra before discrimination and to further investigate ANN to see if it really is applicable to our studies and if we can get it to work faster. As well, for future work, we want to see if ultra-pure water can lower our background signal to improve our detection, and we want to try and make our bacterial deposition more uniform. We would also like to implement pre-processing techniques prior to discrimination and continue working with silver microparticles as we've done in the past to enhance our emission spectra. Thank you.